Staphylococcus aureus for my project presentation to research, and my name is Keely O'Brien. Some background information on Staphylococcus aureus is that it is an opportunistic pathogen. It is also gram-positive, and it is the common cause of skin and soft tissue infections, as well as other invasive infections that can result from surgical sites and medical devices such as catheters. It's acquired mostly through the community, through like direct contact or skin-to-skin contact from surfaces, as well as hospitals. Yes, and then it has Cauchy morphology, so it's little balls, and it is a facultative anaerobe, meaning it can survive in both with oxygen present and without oxygen present. Without oxygen present, it just uses fermentation, and with oxygen, it's going to use anaerobic respiration, and this increases its virulence because it is versatile in where it can survive. Staphylococcus aureus also is resistant to antibiotics, and this presentation will focus mainly on how it uses signaling, gene regulation, and host cell manipulation to survive treatments, antibiotics, and host cell immune recognition, and ultimately to establish chronic or acute infections. Transmission and acquisition, direct via skin-to-skin contact, contaminated surfaces that have not been disinfected, entry through open wounds, a lot of the times through like surgical sites. If the instruments are not cleaned properly, sterilized properly, then this could likely result. And you do have an increased risk if you are immunocompromised, meaning if you are very old or very young, you could be immunocompromised and at a higher risk. And overuse of antibiotics has led to antibiotic resistance, and medical devices such as catheters and implants, which I will go into further detail on. Signs and symptoms include skin abscesses and boils, infections from open wounds or surgical sites, localized inflammation and fever are systemic symptoms of the host cell trying to fight off this chronic or acute infection, as well as pus formation. So all of these will occur due to host immune activation. The Staphylococcus is actually the leading cause of surgical site infection as well as medical device infections due to the biofilm that develops, and it allows the bacteria to remain persistent regardless of treatment. Virulence regulation matters because virulence is understood to be a dynamic regulation rather than a static one, meaning there's multiple complex signaling cascades and communication that increases the virulence of Staphylococcus aureus. It's regulated by metabolism and signaling pathways, and later you'll see that there's multiple sensors and gene regulation through operons and repressors that will be expressed or repressed given certain environmental conditions that might or might not be advantageous. Virulence permits the adaptation to host cell environments, allows the bacteria to evade host cell immune recognition. So the first journal I did was on biofilm formation by Staphylococcus aureus is triggered by a drop in the levels of a cyclic dinucleotide. So more specifically, this study studied what role that cyclic diadenosine monophosphate played as a secondary messenger, and it works intracellularly to create or regulate extracellular biofilm through signaling pathways and quorum signaling. So lower levels of this cyclic dinucleotide led to increased biofilm formation, and this secondary messenger was also responsible for altering adhesion strength and biofilm matrix production. So when it was not advantageous for the biofilm to have a certain structure, like be in a certain place, it would simply just like adapt to that, and it could change it. So it was a dynamic system. Biofilm formation increases antibiotic resistance. Antibiotics can't target through the biofilm. It has reduced metabolism, and it is adaptive in this Staphylococcus. The next journal I did was on Staphylococcus aureus suppresses the pentose phosphate pathway in human neutrophils via the adenosine receptor to enhance intracellular survival. So we've already learned about the pentose phosphate pathway. So it survives intracellularly through manipulating host cell immune metabolism. And this study was actually very fascinating. So it targets these receptors in human neutrophils, and it activates these adenosine receptors which suppress the pentose phosphate pathway, which directly is going to decrease the NADPH production. So remember, that's the electron donor. And then without the NADPH, the production of the reactive oxygen species, which is responsible or helps kill off this infection, is not produced. And suppressed reactive oxygen levels lead to impaired neutrophil antimicrobial killing. Okay, and then the next journal I did was on Staphylococcus aureus. RNA3 binds to two distant regions of CoA, which is a gene. It's not a gene, it's an enzyme. And then before that enzyme gets produced, it attacks the mRNA to arrest translation and promote mRNA degradation. So RNA3 is the effector of the AGR signaling complex, and the AGR signaling complex is the accessory gene regulatory complex, and it is responsible for quorum signaling, which is like how biofilm communicates. And RNA3 directly binds virulent mRNAs. It blocks translation and promotes mRNA degradation. So at the translation initiation site where the ribosome would attach to translate the mRNA, it blocks that. So that's how it degrades the mRNA. It's involved in checkpoints with virulence expression. It also acts as a post-transcriptional regulator, and it ensures virulence is only expressed when it is advantageous for population density and host conditions. So if, say, for example, glucose was low, then this signaling mechanism in this RNA3 complex would bind and regulate like certain gene expression, maybe conserve like nutrients or make the biofilm smaller in order to remain alive within the host cell. And micrococcal nuclease, well, this was the next journal that I did, was on micrococcal nuclease, and it stimulates Staphylococcus aureus biofilm formation in a Maureen implant infection model. So enzymes secreted modify extracellular DNA, and this extracellular DNA is responsible for a lot of the structure and stability that's associated with the biofilm. This micrococcal nuclease promotes biofilm stability in vivo, and nuclease was expected to inhibit biofilm stability, but this study actually proved the opposite. We know that nuclease is responsible for like splitting things and fragmenting things, and we thought that more of that kind of degradation process would cause the biofilm to be less stable, but it is adaptive and dynamic, and it actually increases the virulence. Biofilm on implanted medical devices is more persistent with nuclease. However, or because nuclease is constantly adapting to new environments and taking away some of the stability of the biofilm, adding some back in order to increase its stability. And the degradation of extracellular DNA restructures biofilm to be more persistent through improved cohesion. And more stable biofilm leads to less host cell immune response. Example of a medical device that is at risk of infection are heart valves, heart valve implants, also like a knee replacement or like intervertebral discs, like a herniated disc replacement. Anything that's made of like these biomaterials is at risk for this infection. Also because Staphylococcus is facultative anaerobe, so it can survive just about anywhere. And then the collective application of this essentially is saying that the dynamic system integrates processes such as metabolism, signaling, and gene regulation, all to increase its virulence. And biofilms lead to chronic and acute infections in human hosts. Interactions between host and pathogen are important to developing effective antibiotics. And virulence is an adaptive and tightly regulated system. And with that understanding, antivirulent therapeutic treatments can target specific pathways to prevent infection. Each of the 4 studies that I did on this, their conclusions alluded to a promising future for antibiotics and potential other approaches to mitigating this severe infection. And these are my references. Thank you.